All right, we're going to resume. Everyone, if you want to come in and sit down. Okay. Did you drive here? Did you fly here? What's that? Okay. Well, they're going to miss good stuff. So, everyone, thank you. Looks like we have a little hoopla outside, so hopefully they'll be filtering in. Our next program is actually from the University of Minnesota, and our first speaker is Dr. Chester Whitley, and I'm going to let him do some introductions of himself, but thank you so much.
Well, thank you so much, Cindy, and thank you all for attending. And that's my clock, isn't it? Anyway, I guess I'm on time. So I want to thank Cindy also for being a member of the NIH-funded Lysosomal Disease Network External Advisory Committee. So we've been running for about 12 years now, funded by NIH to do natural history studies to help create the fertile soil for actually doing clinical trials. Cindy's been a long time member of the rare disease community, certainly, and it's a real honor to have you all here and to be able to discuss with you some of the things we're doing and so forth. It's really exciting to see how big a meeting you have and how many people here. Thank you, Cindy. So I want to go back here to the inspiration of Peter Rosenthal. This is what I'll talk about. He is the man who tried everything, and specifically to treat Gaucher disease. Now, I met Peter maybe 15 years ago, and he was sort of a middle-class person in doing his family history. One of the questions we always ask, and this is relevant to his last name, are you Jewish? Just so many people are Ashkenazi Jewish in their heritage. And Peter was absolutely adamant about a lot of things, including the fact that he was not Jewish. But, everybody has their own perception, and maybe he's right. Apparently he didn't practice actively as a Jew, but his name was suggestive anyway. But he had Gaucher disease. When I met him, he had a wonderful girlfriend. He was self-employed, maintaining a couple of condominiums and so forth, and just so darn energetic. I remember coming to clinic one time, and he was telling me what he was doing that day. He was knocking the ice off of his roof and repairing this and that, but a tremendously active man, and this included through his advanced age. And he survived quite a long time. So in the discussion here, I want to thank several people who contributed in various ways to what I'll say here. Dr. Janine Jarnes, who's a doctor of pharmacology, she writes all of our prescriptions, and she does amazing and creative things to make sure people can get their therapy, both getting insurance approvals, but making sure they're able to stay on therapy despite reactions and all sorts of things. Also, Michael Prisbill, he works in our laboratory, and I'll show you some of the stuff that he's doing. I will say we're at the University of Minnesota, so it was a pretty quick drive over here in the Experimental and Clinical Pharmacology Department, College of Pharmacy with Dr. Cloyd here and Reena Kartha, and the Gene Therapy Center at the University of Minnesota. So before I get into that, I want to plug something else here. For 19 years here, we've been running something called the World Symposium. World means we're organizing research on lysosomal diseases. It's evolved over time. The very first meeting 19 years ago was just down the street here, about 5 blocks, in the Marriott Depot Hotel. It was great. It was funded also by NIH. There's a long story on that, but it continued on, and we're meeting just about every February someplace warm, because I like to get out of here, and it'll be February 22nd. So patients are welcome to attend. It's intended for physicians and researchers. However, you get in free if you're a patient. So go on the website and check it out if you're so interested. We're organizing research on lysosomal diseases. This is actually a light microscope. The logo is a light microscope out of a brain cell. And the red dots are where we've put in the normal enzyme in a mouse. Just a micrograph of the brain. It's been our logo. Well, back to Peter. Peter was born in 1957, and he passed away in 2018 Often, most of the time, all the time, we don't use patient names, except in a case like this where he's explicitly said, I want you to talk about me, and where he donated his body to science when he passed away. But about his Gaucher disease, I met him when he had finished one of the clinical trials that led to the first systemic treatment for a lysosomal disease by a pharmaceutical company. The first lysosomal disease treatment we did at Minnesota, 1983. It was inspired, 1983. It was actually inspired by a group in London. But we did bone marrow transplants to determine if bone marrow from a donor would not only correct the disease by making enzyme and circulating around the body, but also answer the question, did it get to the brain? Did the enzyme coming from the donor's bone marrow get across the blood-brain barrier? And frankly, to my surprise, 10 years later, it turned out to be true. The next innovation in therapy for lysosomal diseases was that by Bill Gall. And at the NIH, he developed the first FDA-approved drug for lysosomal disease, cysteamine. And that's now available as eye drops. And then after that, Genzyme came along with, now known as Sanofi, came along with the first enzyme replacement therapy. And Cindy, I think, knows that very well. And perhaps, she's nodding, I can say more than that. Cindy, I've got to ask you, were you in the first enzyme replacement trial with placental-derived enzyme, or the next one? She was. So that drug got... Late 80s, it was approved by the FDA in 1991, the first pharmaceutical systemic therapy for any lysosomal disease. There's a pioneer right there. Peter was in that same trial, although I don't think they knew each other. So he was on that trial of Ceridase, and then when Genzyme, then called Sanofi, now known as Genzyme, they came up with the enzyme made in recombinant DNA technology, really advanced things. And I took care of people in one or both trials. I have to go back and look, I don't remember actually, here at Minnesota. In any case, the drug was very expensive in those times, and so Peter was always trying to get his dose of drug down to the very cheapest, the very lowest dose, which I had concerns about. It was approved at one dose, and he kept trying to tweak it down so we wouldn't run out of insurance money, which was capped back then at about $1 million. Now we've kind of blown that limit away. But that was his goal. So I was really concerned all the time, but we went along with his desires, and actually was very interested in other therapies as they came along. This was not a wild and crazy guy, but a guy that wanted to see his disease treated well and continue working the way he was. So this shows his history, actually. I encountered Peter somewhere, around here. I think it was the 1990s. And there's a new drug coming out called miglustat. And it was made, around the world, thought maybe it would work for Gaucher disease. And in that trial, he had to get off of his enzyme replacement therapy, but continue that oral medication, miglustat. And so what happened is this chitotriosidase was going up instead of staying low. How many of you, if you're willing to raise your hand, are monitoring your chitotriosidase or your chito levels frequently? Very good. At least once a year. I encourage, you don't have to admit to this. This is not a court. You're not under subpoena or anything. There are a few people that are chitoless, yeah, so it's a shortcoming of that biomarker. But anyway, if you can measure your chito, you want to keep it low. That's your chemical blood test, along with TRAP and ACE, that tell you the therapy is working. So Peter had gotten his chito down from probably way up here, down to, about 2,200. But when he went off of enzyme and taking the oral drug, well, it was going up again, not good. So that triggered him when another drug came along under experimental clinical trial basis, taloglucerase, as it was called that then. He started on that and it went down. So that was a good response to that drug. And again, he's trying to keep his dose as low as possible. So I don't know. He kept doing that. But then came along another drug called velaglucerase. And he was taking, he got smart at this point, said, I'll take a low dose of the then FDA-approved drug velaglucerase plus miglustat. Combined therapy. And Janine had a big, hand in doing that. And he was hanging around in here for a while. These are weeks, by the way, up to 99 weeks. You can't read this. And then eventually, as we convinced him to get a lot higher dose of Vpriv or velaglucerase, his levels came down. And now he's getting down into a reasonable range. And if you look at the normal levels, I think it's around 100 in most laboratories, 150. Very few people get their chito down to normal range. So don't be too disappointed if you're not in the normal range, but that would be a good goal. And then eventually continuing on with that medication for quite some time, yet another drug came along called eliglustat, an oral drug, and for him, that worked pretty well. So that was Peter's history up until about here. And then one day he came into clinic kind of staggering and wide-eyed, not knowing what he was doing. And Dr. Jarnes, who had worked in oncology for a long time, said, "I'm very concerned about Peter. I think he's got cancer." Strangely enough, just walking down that hallway, she said, people like that have a low calcium level. She's seen in clinic too many times. It's often a sign of a cancer. We got to check him out right away. In fact, she nailed it. So just measuring his blood calcium, getting him to the oncologist, we found out Peter had one of the complications, rarely, very, very rare, of Gaucher disease, extremely rare. He had another rare, he had a family history of Parkinson's disease. So in many ways, there were other sequelae that you're not always thinking about that he and his family had experienced. Anyway, what's going on now? Well, let me move to the future. Peter was the model for somebody who was willing to try something potentially better. And admittedly, taking a risk for a clinical trial. I will tell you that companies that sponsor clinical trials are going to make money, and they're going to make a lot of money. They have to invest a lot of money to make it happen. And they're not going to fail. In this area of rare diseases, there are very, very few companies, biotechs and startup companies and even major pharmaceutical companies that are willing to lose money and spend the tens of millions of dollars to do a clinical trial. So even though the consent form says you are at risk for dot, 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 and we don't know if it works, dot, 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 all that's true, but as Peter illustrated, more often than not, those drugs are safe, at least their side effects are well defined, and there's a good probability it will work as well as the prior drug and maybe better in some ways. And frankly, my personal opinion, I'm not speaking for anybody in particular, I think clinical trials are something to at least look into very carefully and not be too afraid of them, but maybe make sure you read the fine print if you're consenting into something. Anyway, what's on the horizon? We've already heard a little bit. Right now at the University of Minnesota, not with any corporation right now, we've come up with something called the PS gene editing system. I'll just tell you a little bit about that. PS is just PS. Some people say we're going to call it postscript, but right now it's just PS. But it uses something you might have heard about called CRISPR-Cas9, and it comes off of something done by another company called Sangamo, who did the world's first gene editing. Gene therapy is one thing, and nowadays we more or less think about that as something we do to alter a cell or many cells in the body and put the normal gene in. Typically, it's that kind of scenario that is called gene therapy. However, gene editing is a very specific kind of gene therapy where we send some mechanism in, some machinery in to fix or change a person's own DNA. We're not sending a virus in to just be sitting around for a number of years making the enzyme instead of our own chromosomes, but we're sending in a mechanism of fixing or somehow changing your own existing DNA, gene editing. So this is a form of gene editing. Some people consider that a kind of gene therapy, and you could argue that it is as well. So in any case, we've published in our lab these publications looking at other diseases, not Gaucher disease. So mucopolysaccharidosis type 1. This is all mouse work, by the way. A disease called Tay-Sachs-Sandhoff disease, and most recently, something else called GM1 gangliosidosis. By the way, most of those have a very heavy-duty brain component. So we started doing some work in our mice models for the company Sangamo. We found that it worked very well in our mice that have Hurler syndrome, MPS 1, or Hunter syndrome, MPS 2, and they took it to clinical trials. It did not work as well as they hoped. It was safe, but the amount of virus or vector we had to put in to do the gene editing was not enough to really get as many cells gene edited, and therefore did not make enough enzyme. But I'll show you a little bit of background behind that. We're still pursuing our version of that, which should be more efficient. So if you're used to looking at DNA maps, this is what you're looking at here. We show here how the CRISPR-Cas9, here's the Cas9 enzyme, puts a gene or cuts the DNA right here in your normal albumin gene, right between what we call exon 1 and exon 2. Now, I'd be a whole couple hours explaining what an exon 1 and exon 2 is, but it's inserting it into the actual albumin gene you have. And it's going to make an enzyme at that location. And so we use something called homology-directed repair or non-homologous end joining. Anyway, we insert the therapeutic gene. If this was Gaucher disease here, this would be the Gaucher gene. We put it right here, right at the beginning of the albumin gene. So now your liver, at least a very, very few cells, think they're making albumin, but they're really making the Gaucher enzyme. And it's coming right from your liver and only in your liver. So when we do that, you can see here the strategy. And the reason we chose liver, partly because liver is very easy to get to. It's an ideal target for tissues treating genetic diseases because the hepatic or liver transgene, the one we put in expression, is known to induce tolerance to the immune response. So even though it's a foreign gene, your body has not seen normal enzyme, the body helps create tolerance of that over time. It's a generalization that we think of when we talk about the liver. Also, the clinical experience with liver gene transfer is out there. People have done a fair number of gene editing in liver cells. We have a pretty good idea about what the effects are if we do it right. And it's a highly vascularized tissue. Therefore, there's a lot of blood vessels in the liver. And it's therefore easy to get the enzyme out into the blood from the liver and secrete proteins. And you could do Gaucher disease the same way, which is why I bring it up. We're not doing Gaucher disease, but it'd be easy to do. So here's some data that kind of Sangamo started with and then we elaborated upon. The normal enzyme replacement dose for Gaucher disease would be this amount of milligrams of protein for an average 75 kilogram person. This is the way pharmacies often think about a body size. It's all adjusted per weight. So if we take what's called the average human being, it's called a 70 or maybe 75 kilogram person. When you get your drug, you're probably getting that amount of enzyme, 50 milligrams. And there's the half-life. How long does it last in the blood? 3 to 10 minutes. And then what percent of the total albumin rate, what amount of your normal albumin would we have to capture and convert to enzyme by gene editing? So it's a very, very tiny amount, just less than 1%. So if we captured just a few of your albumin, just a few of your liver cells, gene edited them to make the therapeutic enzyme instead of albumin, we'd have the equivalent of ERT. One of the advantages is you're getting the ERT not just every week or every other week, you're getting it every day, every hour, every minute. It's continuous, like your body had intended, so to speak. So those are the reasons. Here's what we do in the animal, in the laboratory, maybe in the human. We first package the elements of the CRISPR-Cas9, the guide and the Cas9 enzyme, plus the therapeutic Gaucher or Hurler enzyme in this case, put into 2 adeno-associated virus vectors, commonly used vectors. There's over 120 clinical trials using AAV vectors. They're relatively safe, as we know, at least so experienced so far. We give it intravenously one time. In this case, we give it to the tail vein of the mouse. You give it in the arm one time in a human being. And the therapeutic DNA is permanently inserted into the albumin gene or locus of the liver. And then those liver cells start secreting the therapeutic proteins just as though you were getting them from an IV infusion. The previous studies in terms of neurologic disease were very interesting. I'll segue this. It may not be important to you unless you're thinking that maybe good treatment of your Gaucher disease would reduce your risk for Parkinson's disease. That is purely theoretical right now, purely theoretical, but that's one of the things that's been thought about. In any case, we want to look at how we would potentially impact both the rest of the body and the brain, so we get a high and constant enzyme production within the liver that goes to various places, and it can go in the brain. Now, most people say no. We submitted an NIH grant that said we want to do this and treat the brain disease of Hurler syndrome. One of the reviewers said, well, I just don't believe your data. There are people that cannot get around dogmas that enzyme cannot get from the bloodstream to the brain. But guess what? Beginning in 1983, we did this for Hurler child, and they are now cognitively at the level when they came in. A couple of weeks ago, I saw a patient I did a bone marrow transplant for with Hurler syndrome who was 40 years old. Kids with that disease were usually severely mentally damaged. They died between 5 and 10 years of age. Kathleen, who I saw, was patient number 2, got a bone marrow transplant, and her cognitive level is where it was more or less when she got her transplant. Not good because she'd already had advanced brain disease, but that's the intended process. These are animal studies illustrating that you can get enzyme across the blood-brain barrier if you just give enough. It's usually very, very high levels. So you can look at some mice studies here, mice with alpha-melanocytosis, MLD, aspartoglycosinuria, Krabbe disease, MPS 1, MPS 2, MPS 3A, MPS 7, guinea pigs that have melanocytosis, dogs that have Hurler syndrome or Hurler-Scheie syndrome, mice that have Sanfilippo, Pompe disease, MPS 1, MPS 2. All of these published studies talk about if you give an animal enough enzyme, you can get it across the blood-brain barrier. And so that was one consideration. So I don't want to spend a lot of time on this because this may not be as relevant for you, and I am running out of time. Dr. Kartha's got some discussion to do, but these are mechanisms that people hypothesized would get enzyme from the blood, regardless of where it's produced or injected, if you give enough. And there's other things that we would want to do to get the FDA to agree this is worth doing in a human being. In other words, launch an IND, investigational new drug protocol to start trying PS gene editing. It's not yet been done in a human being, but we're looking at that possibility. So Dr. O has published this that we make reference to here. I'm going back to our MPS 1 example. This is Kelly, the first child in the US, transplanted here in 1983, actually September 3rd, 1983, who did pass away at 16 years of age, but had a developmental quotient equivalent to what she was when she came into transplant. And so with that in mind, the scientists and the FDA will look at various doses of drug. Whoops, sorry, I'm going too fast on that one. And we look at various things like, this only goes backwards, only goes forward. No, my finger's too fat, that's all. So the animals had no toxic effects. We get things that are illustrative of what is happening, such as there's no antibodies or limited enzyme made against the enzyme itself. There are some, but not to the Cas9 part, and a little bit to the AAV vector. Those are things we sometimes give immune suppression for when we do this kind of intervention for a short period of time. We look at other indicators of immune response, and they're acceptable, although systemic steroids would probably be given for a short period of time, like a month. And we also look at plasma, ALT, AST liver function enzymes, creatinine, albumin, the kinds of things the FDA would be concerned about you look at anytime you do a clinical trial. And in summary, we thought this was a very highly efficacious approach. The gene editing leads to a significant amount of enzyme activity and reduction of the bad stuff, the storage material, and that it was seen to be well tolerated by the mice. So in summary, thanks everybody who participated in these studies. They're not published, they're available if you want to look at them. And again, a reminder, maybe I'll see some of you at World Symposium. Thanks very much. Dr. Kartha?
So we'll let Dr. Kartha speak, and then we can have questions for the two of them. Dr. Reena Kartha is Assistant Professor and Associate Director, Translational Pharmacology at the University of Minnesota College of Pharmacy. Her lab focuses on deciphering the pathophysiological role of oxidative stress and inflammation in rare disorders occurring due to inherited error of metabolism. Their research includes the identification of novel protein or mRNA-based biomarkers for early diagnosis, prognosis, or treatment response using patient-derived samples. And Dr. Kartha will let you know what that all means. So.
Thanks, Aviva, for the introduction, and thanks to GCA for having me here today. So I just wanted to take the microphone. Okay. Okay, so is it better? I have to see my slides too, so I need to. Yeah. All right. So I just wanted to bring in a different angle for this disease. That it's not just a lipid storage disease. I think that this morning there was a panel talking about the mutation and the storage and the GL1 and all the lyso-GL1 and all that. So what I wanted to convey is that what we know is that Gaucher disease is caused by a mutation in a gene, GBA1, that leads to a which usually makes a glucocerebrosidase enzyme, but in your case, because of the mutation in GBA1, the enzyme is not made properly, which leads to the accumulation of toxic lipid substrate. Now, what may be happening, and the current treatments is extremely really good. What we are trying to do is replace the enzyme that is defective, so that is enzyme replacement therapy, or prevent the production of the lipid substrate by the substrate reduction therapy. And I know you all know more than me about this. I learn from you all the time. But what may also be happening is that you have this defective enzyme, which is causing misfolded protein. The protein is not made properly if your mutation is not a null mutation, like you're not completely knocking off your gene. There is some amount of protein that is made, but that protein is not folded correctly. And in a good way or not, our cell has a property called unfolded protein response. It tries to correct that mistake by trying to fold the protein into the right conformation or getting it to the right organelle where it can do its job. And in that process, it can trigger other disease pathways. And I think there was a lot of discussion around inflammation. And what I wanted to focus on is oxidative stress, which is basically an imbalance in the amount of toxic chemicals that one would accumulate. And it's actually, in a human or a healthy cell, you have a balance between the antioxidants and the toxic radicals or toxic chemicals. But when you have oxidative stress, you basically tip the balance. So you tend to have more toxic chemicals and less antioxidants. So again, this is another depiction of that. So you can see that there is this misfolded GCase, which is trying, and UPR is the unfolded protein response. So within the cell organelle, it's trying to make the protein in the right conformation. And in that process, it uses a lot of glutathione, which is one of our primary endogenous or antioxidant. That is something that our body makes on its own. And when you're trying to fold the protein correctly, you're using more of the glutathione than what you make, which can cause a reduction in antioxidant. And that can be one of the reasons for the lot of comorbidities associated with Gaucher disease, including the different types of cancer or Parkinson's disease, et cetera. So when you have oxidative stress that is harmful to your body, and increasing glutathione may be an approach which can kind of mitigate some of these downstream consequences of oxidative stress. So we did a preliminary study with about 30 individuals. So 17 of them were healthy people, and 13 were people with Gaucher disease who were treated. And we also had 5 untreated individuals. And as you can see, oops, I turned it off. Is there a pointer in here? Oh, yeah, got it. So you can see, compared to the healthy control, the untreated patients, we looked at the amount of total glutathione, which is the antioxidant. And you can see that compared to the healthy people, patients, irrespective of their treatment status, had lower amount of total glutathione. And this is another readout of your endogenous glutathione. And you can see that, again, treated patients are a little better, but they are not within the healthy range. And this is another antioxidant enzyme that our body makes on its own. And this is the healthy controls. And you can see the patients with Gaucher, because of the defective protein that is hanging around, you probably have a lesser pool of antioxidant. And this is a marker of oxidative stress. So when you have these toxic chemicals, it can modify your lipids. And this is lipid peroxidation. It's basically oxidizing your peroxides in your body. And it can also do similar modification of proteins. And you can see that the lipid peroxidation levels are exactly opposite of your glutathione levels. So you can see the antioxidants are lower, while the markers of oxidative stress are higher. So this indicates that there is untreated or treated condition, doesn't matter. I mean, you still have an unresolved oxidative stress associated with your condition. And because of the link with Parkinson, and also that there is also this neuronopathic group of disease, like Gaucher subtypes, we wanted to check what about the status of certain brain chemicals. So we did a magnetic resonance spectroscopy, which is a method of scanning your brain, but you are actually measuring the amount of neurometabolites, chemicals. You can actually quantitate what is in there. And we looked at certain neurotransmitters. So we did GABA, glutamate, and N-acetyl aspartate. These are all validated markers of neuronal health. And you can see, like, if you're looking at the control range, you can see that the patients who are treated, they have lower levels. So these are different regions of the brain. And you can see the values are lower, at least in two regions. You can see the mean values are lower. And this is, again, a small study. It is just a proof of principle, kind of like we can pilot study to show that we can do this method. and we do see certain differences. And we would like to do in patients who are untreated to see if does in treatment at all have any benefit. Because this morning somebody was mentioning about like your systemic inflammatory markers can actually cause more CNS issues or the issues in the brain. And when you are having systemic treatment, you probably are modifying your certain neuroinflammation. We don't know that yet, so we would like to do a study in untreated patients to see, is this pattern similar, or is it worse off, or is it better? We don't know. And we would also like to do it in neuronopathic Gaucher disease. So coming to the new treatment approaches, I think the current treatments are really great. It's actually tackling some of the symptoms, but we heard presentations around gene therapy, where either you can correct the gene, put it back, or you can have the normal copy of the protein hanging around in your cells so that it can take care. But if it is a normal copy hanging around without tackling the untreated or the defective copy, you can have these associated oxidative stress because your body is still trying to fold your defective protein to the correct conformation. So the ambroxol, there was mention of it this morning. This is a pharmacological chaperone which can help, again, fold the protein in the right conformation. We are working on N-acetylcysteine, which is another antioxidant. There is also studies on CoQ10, which is an antioxidant. This is arimoclomol. So all of this are trying to take care of the misfolded protein. So it's basically you're not fixing the mutation that you have, but trying to correct the protein conformation so that it can get to the right spot for doing its function. So N-acetylcysteine is something which we have been working on for several years now. So the approved product is acetadote, which is an IV formulation, and there's also an oral formulation, mucomist, which is used for treating pulmonary disorders. The acetadote is used in Tylenol or acetaminophen or paracetamol overdose for people in Canada, and it has an orphan drug designation. But we are working with the over-the-counter supplement, which is available as an effervescent tablet or a capsule. And N-acetylcysteine is primarily a source of the amino acid cysteine, which is one of the starting component for making the antioxidant glutathione. So if you don't have glutathione, you can basically use NAC as a source for glutathione because it will make the cysteine to generate the glutathione, and thus it would be a safe, effective, and low-cost antioxidant, which is shown to have some anti-inflammatory action, but it is not proven. Again, preclinical studies show that it may have a benefit. We'll have to see how it works. So this is the study that we are involved in, or we are conducting, which we had to put on hold due to COVID, and we are planning to start again. So idea is to follow the patients for 3 months at baseline, collecting blood samples, and at the end of 3 months, do a magnetic resonance spectroscopy, then give the product Pharmanac, which is the effervescent tablet, for 90 days, and collect blood samples, follow them again during the 90 days, and finally do the end-of-study visit after 3 months after that. And we want to do a battery of oxidative stress markers, inflammatory markers, as well as look at the concentration of NAC, which we have an assay to measure, the levels of NAC as well as glutathione, so that we can basically correlate what we see with the drug concentration to see if there is enough exposure, and is that why what we are seeing, the response we are getting. So these are the participant characteristics. So far we had enrolled 13 individuals who are treated. And you can see they're more or less similar age as the control population. It's only for the baseline assessment. They are not getting N-acetylcysteine. We have these different mutations so far that are represented in our cohort, and almost equal split between ERT or SRT. And if you look at another assessment we are doing is to kind of measure something that is relevant for you all. I mean, something which is important for you. So we adopted like the brief pain inventory and the fatigue inventory to measure the amount of, to assess the level of pain and fatigue that individuals are experiencing in spite of being on treatment. So this is just a representation of the average pain that people experienced. So about 60% said they have mild pain, and about 30-40% on moderate. And with fatigue, there is a greater proportion, around 70% said they have mild to moderate fatigue, and 30% on moderate to severe fatigue. And about 50% were on some kind of analgesic to control their pain. So this is our preliminary results from the interim analysis that we conducted, and I just wanted to show some of the parameters here. So look, if you look at the blood, total glutathione or catalase, this was the data I showed before. The baseline levels were considerably lower compared to the healthy. And with NAC, it seems to be improving. It did not reach clinical significance. That doesn't mean anything, because if you don't feel anything, then having a p-value of less than 0.05 doesn't make any sense. But as you can see, there is a lot of variability. There are few individuals where it increased really well, and there are few that it has decreased. And same thing with catalase. And if you look at the brain glutathione, we did not see, if you look at the mean value, there was no difference. But if you look at the individual values, there were few that came down, but there were others who were responding, where the glutathione levels were increasing even in the brain. And this is following an oral treatment. So I would like to add here, earlier we did a pilot study where we gave a big dose of NAC as an intravenous infusion, and we were able to see a 30% increase in brain glutathione. And that's why we embarked on this study. But we don't see that kind of increase, but we do see some trends here. But most importantly, about 38% of the patients said they experienced a mild improvement in their average pain, and about 60% said they're feeling much better. Their brain fog is lifted, they're feeling more alert. And we don't know if, we need to figure out if these are the same patients who are showing an increase, but we would really like to see what their level of NAC exposure is and trying to correlate with what they are seeing here. So we are continuing enrollment, and we would like to try with a little higher dose just to see what the benefit is. And one other thing which I wanted to kind of share is that when I was presenting this data, there was a question about, are we seeing anything improvement with the level of enzyme? Does it act like a chaperone? Because these are small molecules. So we did in vitro studies using patient-derived skin cells, and we tested both NAC and its metabolite cysteine, and we do see there is an increase in enzyme activity, but this is in vitro. But seeing that report, a physician came, I think she prescribed or had some people take NAC, and she shared the reports with me, and these were all carriers, individuals with just one copy of the gene. One of them had the mild mutation N370S, and two had the null allele. You can see that the individual with the mild allele had a 67% increase in GCase activity. This was a lab test that they had sent out with LabCorp and done. While those with the null allele, you had like a variable response, which may indicate that even with that wild type allele, they probably have some mutation which is not known or something. They may not be a full carrier, or it's something more to it. So it says that the chaperone effect may be mutation specific, and we need to do more studies on this, basically using this enzyme activity as an outcome measure. Now, I just wanted to highlight some of the studies that we are currently ongoing. We are enrolling participants for this. So we had baseline, initially we did that baseline assessment of oxidative stress markers, and we would like to expand it to study more inflammatory markers. And we are enrolling 10 untreated and 5 treated patients. Again, this involves just blood draw. And earlier we have been working with a remote phlebotomist who would work with you to get the samples. With COVID, I'm still trying to figure out the logistics, but the study is open. We are also interested in characterizing extracellular vesicles. So I think I just wanted to quickly summarize. I'm sure that you all know that the lysosome, the organelle that is defective in Gaucher disease, is acting like a trash bag. And the way it eliminates trash is through these small vesicles, like little trash bags that they throw out. And this can be of different types. And what we hypothesize is that when you have a defect in the lysosome, the type of trash that is thrown out and the bags that are carrying it may also be different, and the extent to which the bags are thrown is also different, the number, the quantity. So we would like to characterize that, and for which we are enrolling patients who are untreated for their Gaucher, 10 obligate carriers, and 10 healthy volunteers. And this involves 2 blood draws, where we would utilize the blood samples to do a genetic testing to make sure where the mutation is, and do the characterization of the extracellular vesicles, which is the trash bags. And finally, we are also doing that what I mentioned initially about that MRS study in patients with neuronopathic GD. So this is a type 3 GD. And this is just a very small study trying to see, okay, we saw some differences in type 1 GD. Is that really true? Well, how does it look for type 3? And we have the protocol, so we would like to run it at our center. And we will be enrolling 5 patients with GD3, irrespective of the treatment status, because the current treatment, not the venglustat, but the current ERT will not cross the blood-brain barrier, and so we would like to enroll patients for that. So if you have interest in participating in any of the studies, or if you would like to refer to somebody, please do that. And I would like to thank Dr. Cloyd, who gave the introduction about our center yesterday, and he has been the PI on the NIH, the LDN study, which was the NAC study that I mentioned, and other collaborators and students and postdoctoral fellows working in my lab, and also the funding from Sanofi, Pfizer, Takeda, and all participants who have helped me with this study. Thank you. Obligate carrier is when there's one copy of the mutation. Yeah, I just used this fancy name. That was a fancy word. Yep.
Is there any problem with taking too much NAC?
It can have a problem. We don't know. That's why we want to do a higher dose. I mean, it's not like a linear increase. I mean, we don't see a linear relationship. If we keep increasing, we don't see the benefit increase accordingly. This is another group of patients that we tested NAC. So we don't know what is the safe limit.
What was the NAC dose on that trial, the first trial?
The first trial is 3,600 milligram.
Well, now it's 36. The first one was as well?
Yeah, what we did is 3,600 milligram. I mean, it's relatively safe, so I don't think there is an overdose issue, but if you take too much, then maybe there is some issue. I mean, it can cause gastric problems because it's a big load of sulfur-containing amino acids, so. Yes?
The title of your talk was that Gaucher's is not just a lipid storage, but that there are other disorders, and you mentioned cancer and Parkinson's. Are there other ones that are interesting you that you're looking into?
So, I mean, I'm not looking into those other comorbidities, but what I'm interested is in this pathway that can lead to these other comorbidities. I'm sure Dr. Mistry's lab has extensively studied multiple myeloma, MGUS, and all that, and the inflammatory pathways. So, I'm not specifically looking at any comorbidities as such.
But besides Parkinson's and cancer, are there other things that you think it leads to?
I'm going to deflect to Dr. Mistry. I mean, are there other than Parkinson's and cancer and MGUS? Are there other comorbidities that are associated? I know skin issues are there, right?
So we are beginning to learn something new every day about Gaucher disease. And what I'm happy about is that I think it gives us greater power to ensure that we are monitoring our patients. So Dr. Sibransky talked about Parkinson's disease, and she is always very compassionate and understanding that this is frightening, but the risk is very small.
Mm-hmm.
And so this is what's happening with a lot of, some people call it comorbidities, but they are direct results of this lipid buildup and inflammation. And in some individuals, that's what it is. So if you look at the disease trajectory in every patient in this room, you're going to find unique, what we call phenotype signature in these individual patients. And so one of the holy grails of medicine is to identify genetic modifiers. And there are two schools of thought. One is that you're going to find one additional gene, the variants in which will interact with Gaucher, and it will explain all of the genetic modifiers. That's not going to happen. I think that, we have more than 20,000 genes, and these networks between the Gaucher disease pathway and all the other genes are very, very extensive. So just as an example, thankfully we don't see many patients nowadays with a devastating disorder called pulmonary hypertension. It is life-threatening, and it occurs with very high frequency, especially among people who had their spleens removed. This is not a comorbidity. It is wrapped around that patient's individual genome variants. And what we find is that patients who have variants in a pathway called ESPR2 pathway have a very high risk of having pulmonary hypertension when they also have Gaucher disease. conversation